Hi, welcome to UCLA. My name is Jimmy Hu, and I am an assistant professor in the MBIDP program. Our lab is pretty new, just over a year old. There are currently 4 people in the lab, including myself, and we're always looking for new graduate students who are interested in developmental and stem cell biology. Our lab is interested in answering 2 important questions in the field. The first is to understand how an organ is sculpted into the correct shape during development. While the second is to understand how stem cells are regulated for tissue regeneration in adults. In our lab, we utilize the craniofacial structures as model systems to study these questions. During development, many of the structures from within the vertebrate head develop from a single layer of epithelium. These cells then undergo complex morphological changes to arrive at their eventual morphologies. And so in our lab, we're interested in understanding what are the genetic, cell biological, signaling, as well as biomechanical regulations that control this process. And we utilize many different techniques to study these questions. We utilize mouse genetics, and we conduct live imaging to look at cell behavior. We also use organoid culture to answer important questions at on a more simplified platform. And lastly, we utilize biomechanical approaches to ask the questions of how tissue mechanics may control morphogenesis. And one of the questions that we have become very interested recently is how different organs are defined in their specific developmental field, and how boundaries are defined during this process, such that the organ will only arise in specific spots during development. And so to begin to address this question, we have recently conducted a single-cell seq experiment using cells from the epithelium as well as mesenchyme from in the mandibular in the embryonic mandible. And so here I am showing you some of the pictures of the gene expression within the epithelium. And we have identified several subpopulations based on the clusters that are in the single-cell seq experiment. We have found genes that are specifically expressed in teeth, for example, or cells surrounding the tooth, as well as complementary expression pattern that outlines the tooth. We found expression in the tongue. We found expression in the overlying epithelium, as well as other interesting expression pattern. And we're amazed by how these genes are specified in their own field. And we're in the process of trying to understand their functions and how they're utilized to establish different patterns. And so one of the potential rotation experiment is to look at our data in the mesenchyme. Remember, the last slide only showed us the genes expressed in the epithelium. And so one of the potential projects will be to look at the mesenchymal data and to begin to identify some of the mesenchymal subpopulations and study the functions of candidate genes in patterning the tissue. We also want to correlate this data to our effort in understanding how tissue mechanics may control gene expression. And so a second potential project will be to be part of this collaboration with Dr. Ujay Campus at UC Santa Barbara to use the oil microdroplets as a force sensor in order to measure tissue forces in different parts of the mandible. We will then be able to match these different forces into differential gene expression in the developing mandible. And this will be a start to start understanding how tissue mechanics may potentially control different aspects of cell biology as well as gene expression of specific genes. And so these are some of the developmental projects that we have in the lab. And at the same time, we also study regulation of adult stem cells in adult organs, including the mouse incisor. So the mouse incisor grows continuously, and this is because of maintenance of a group of adult stem cells in the tooth. And we have previously found that YAP, the transcription cofactor in the hyposignaling pathway, is absolutely required for maintaining cell proliferation in the adult stem cells. And we have also found that when there is an ectopic increase of YAP activity in the more differentiated cells, these cells then resume proliferation, and we suspect that they may have been converted into a more progenitor-like state. And so a third potential rotation project will be to further look into this and think about how YAP may convert differentiated cells back into a stem cell-like state. And if this is true, then this could potentially hold regenerative potential for treating patients and derive new stem cells. And so here are some of the examples of the projects that we do in the lab. These are people in my lab. And if you're interested in any of these projects, feel free to get in touch with me, and I'll be happy to talk to you more about these, the questions that we're interested, as well as projects in the lab. Okay, thank you, and bye-bye.